Welcome back, everyone, to Inflammatory Content, the podcast all about immunology. I'm your host, Kellen Cavanero. Today is a big day. We are going to do something different, something I've been wanting to do since I started this podcast, and that is to record a conversation with a top researcher about their science and journey. Our guest today is Dr. Alan O'Neill. Alan is a microbiologist, immunologist, and also a friend and colleague. He is currently a project scientist in the same lab as me, and that is Dr. Richard Gallo's lab at the University of California, San Diego. In this episode, we talk about how Alan became the successful scientist he is today, his work on microbiome therapeutics with a focus on his recent publication in eLife. You can find the link in the show notes. We also talk about imposter syndrome and much more. A word of warning. This was my first time ever recording a conversation for the podcast. I started out a bit nervous and realized after we started rolling that I should have prepared my questions in advance. That said, Alan's great speaking ability and scientific knowledge made up for my shortcomings and then some. So without further ado, please enjoy this conversation with Dr. Alan O'Neill. Alan, welcome to the show.
Thank you very much.
It's good to have you here. I've been wanting to do this conversation for a bit, so I'm excited to get to know a little bit more about your background and hear about the paper you recently published. So I think kind of the first question and logical place to start is, what got you into science?
I think what got me into science probably was an image of a bacteriophage in my first year high school biology class. I wasn't really all that interested so much in school or even biology, but I remember going through the textbook and I remember seeing this really striking, very cool image of a bacteriophage sitting on the surface of a microbe. And I remember thinking that this was just like the coolest looking thing, like the capsid of the bacteriophage looked like something kind of out of the Matrix, very kind of sci-fi. And I always remember that captured my attention. I was fascinated by that.
Where did you end up going to college doing your undergrad?
I went to UCD, which is University College Dublin. It's a huge college. I think it's the biggest college in Ireland. It's on the south of Dublin, so I'm from the north of Dublin. So it was kind of a big trek to get there. It was a big decision to make to go there. I had to get basically 2 buses to get to the campus. 2 buses there, 2 buses back. So, every day I was traveling for maybe an hour and a half. But I really wanted to go to that campus. I really wanted to go to that university.
Did you end up doing some undergraduate research there too, or did that not come until later?
That didn't come until later. Yeah, I guess maybe there wasn't so many opportunities to do undergraduate research, or again, I never really considered it so much. The only time I really got into a lab and got my hands dirty was in my final year. And the first semester was just a dedicated research project, and that was around 3 months. That was the first time, apart from the practical classes and stuff like that. This was the first time I was in a lab properly full time for several months doing a project. I did a project on HTLV1, and I had a supervisor, Dr. Norian Sheehy. She was a great mentor. She inspired me a lot, and I think doing that project really stimulated my interest in research. I think that's what really made me want to go on actually and pursue research as a career was that project that I did.
Did you have an opportunity to work with a postdoc, or were you kind of doing your own thing?
Surprisingly, I was directly mentored by Dr. Sheehy.
In the lab?
In the lab, yeah.
That's nice.
It was really, so she's the principal investigator there, and she has a small group, but she was really, really involved in the research. She loved it. She was passionate about teaching students. And I learned directly from her. She's very good, and she was very, very tough with me. She didn't let me away with much, and I learned a lot from her. She was a great mentor and inspired me a lot. So that's what definitely made me want to choose this career.
Were there any particular practices or habits that you learned from her that you've kind of carried with today?
Yeah, I think detailed notes in the lab. this is what she hammered home to me. Anytime she was showing me a technique, Western blotting, anything like that, always had to have a notebook, always had to be writing down very fast all the instructions. I essentially had one chance to learn the techniques and get them right. And the way I did that was by taking extremely detailed notes, and then, of course, following up with other postgrads and postdocs in the lab to make sure that my protocol was correct, and then I would do it.
So you finished your undergrad, did some research. Did you need to get some more experience after that to kind of prove to yourself that you could handle this life that we have for the long term?
I still wasn't quite sure what I wanted to do. So the microbiology degree that I chose actually had quite a lot of industrial elements to it as well. So we had a lot of courses on how bioreactors work. So there was definitely a connection to the industrial microbiology side. I was just more interested in the medical microbiology courses. And so I guess I've never really got very high grades in the first 3 years of my degree, but in the final year I got an A+ on that research project, and I guess I was surprised. I don't know. I didn't usually get a lot of A's, so I think that convinced me, maybe, oh, I'm actually good at this. Maybe I could do this. So then I did very well. I got a very high GPA in my final year, which was my final score. And I think with that, I just thought this is a great platform now that I can go and actually look to do a PhD. I was in the top 3 of my class, really liked the research project that I did. So I thought, sure, why not? I'm going to see if I can do a PhD.
So straight after, did you get in where you wanted to go, or was it a struggle?
It wasn't a struggle. I was surprised. Everybody seemed pretty keen, actually,
Mhm.
on accepting me. I guess maybe it's a little bit different in Ireland versus the US. It's quite common for grad students to come straight out of university into the grad program. The place that I chose was in UCD. It was the School of Veterinary Medicine. I started my PhD there, but I actually didn't finish it as a PhD. I completed that as a master's. I wasn't very happy in that program. I didn't really like the lab that I was in. So this was a very big decision that I had to make, actually. Before I transitioned to official PhD status, I decided not to do that and finished early, wrote a thesis, and I left with a master's. I did that maybe because I was very ambitious. I thought I could do really, really good science. I wanted to go to a top lab, really do elite science. I decided in the end to go to London, and I pursued a PhD in London, actually.
So was the lab you were in just kind of not the right fit?
Yeah, I just don't think it was the right fit. Maybe the atmosphere was a bit laid back. I wanted somebody to push me. I wanted somebody to really kind of make me the best scientist and researcher I could be. And it was a good department. I really liked the people there. My supervisor was good. He's smart. But it's just the department was maybe a little bit too relaxed for me. And I knew I could get a PhD there. I mean, people were very supportive of me and the work was good. But I just thought this was a good opportunity for me to try and go as high as possible. And that was my decision, really. I wanted to be in a very tough lab. Kind of something that's dynamic, high pressure, a group that's really publishing top level science. So I thought I could maybe go for that. I tried, and very fortunate, I did land in a very good lab.
And that was in London?
That was in London, yeah, in Imperial College, London.
And that was what you perceived as like the pinnacle.
Yeah, to me, yeah, to be in Imperial College, it's an incredible place. It's one of the top universities in the world, and the microbiology there is one of the best in Europe. I applied for a Marie Curie PhD scholarship, and I was very lucky to get that. So that's a pretty prestigious scholarship to get. And I interviewed in Imperial College with several labs, And the one that basically offered me the position was Professor David Holden. His lab works on Salmonella pathogenesis. But my project was a little bit different. It was working on group A streptococcus, which is a very different pathogen. So that has its advantages and disadvantages to go into a lab where no one else studies this particular organism. It was a great decision. I loved the lab, and I learned a lot. But it was what I wanted. I wanted a tough lab, and I got it.
Mm-hmm. We had a conversation before about like the process in, I believe it was that lab, about how grad students develop their thesis project. And it was, if I recall, you said it was much more of a independent thing. You're not given a project. You're expected to create the project. Am I remembering correctly?
Yeah, it's correct in a way. I had a specific topic that I had to work on, which was working on the intracellular biology of group A streptococcus. This is what my boss really, really wanted to do. David had spent, 20 plus years working on the intracellular biology of Salmonella typhimurium and learned a lot and published incredible work, but he wanted to take all of that work, all of that insight that he had learned in 20 plus years and essentially apply it to another pathogen, an understudied pathogen such as streptococcus. And so this is what he had planned for my PhD. I had to focus on group A strep, but because no work had been done, I arrived in a lab that didn't even have streptococcus. I had to get the strains. It essentially meant that I had a free role. I could take the project really in kind of anywhere I wanted to go. I would essentially just do very preliminary experiments, doing a lot of optimization steps, doing just infecting primary cells, and then just following whatever interesting phenotype I found. So I had a lot of freedom that way, but it was a specific topic, I would say.
And if you could kind of sum up your PhD in a couple sentences, which is a sad thing to do, what would you say like your major findings were from that experience?
What I found was that group A streptococcus can survive and also replicate in the cytosol of human macrophages. And that's important because before I did that, this wasn't really known actually, whether group A strep actually has an intracellular lifestyle. It was classically seen as an extracellular pathogen. And so there was a couple of observations in the literature prior to me starting the PhD that suggested that group A strep could be found in macrophages retrieved from patients with necrotizing fasciitis, for example. But no one had really studied the interaction of that bacteria with human macrophages. And so that was kind of the novel finding. That was what was really interesting. And I showed a little bit more mechanistic insights. For example, group A strep secretes a streptolysin O toxin. Once inside the cell, this toxin allows bacteria to escape its intracellular vacuole. And so it escapes from its vacuole into the cytoplasm with this toxin. And then once in the cytoplasm, that's obviously a really nutrient-rich environment, and the bacteria is able to replicate, and that leads to destruction of the host cell.
So after clearly having a very successful PhD, were you pretty confident that academia was the lifestyle and career choice for you and that you'd go on to do a postdoc, or did you kind of have to think about it a little bit?
Had to think about it a little bit. The PhD was very, very challenging for me. It was successful in the end, but during that process, I wasn't sure whether it was going to be successful. As I mentioned earlier, I was ambitious and I wanted to transition to a top lab. And as I said, I got that experience, but it was a very steep learning curve. From the time that I arrived until the time I left, I had matured a lot and I developed a lot. But it was a tough experience at the start. I needed to learn a lot, and I made maybe some mistakes, and I was a bit slow in the beginning. So it was a very tough experience, and my boss was an absolutely incredible scientist, incredibly smart, very, very hardworking, and he was very, very tough on me. Very, very tough. It helped me as a scientist, but also kind of exhausted me as well. And at least in the UK, also it's a bit different than in the US. PhDs are a lot shorter. So with the Marie Curie funding, I had 3 years of funding, and that was it. And in Imperial College, you have 4 years to graduate, and that's it. You don't get an extension. So I had to do this in 4 years. And again, as I said, I didn't even get the Group A Streptococcus strain until 4 months after I started my PhD. So the clock was always against me. At the end, I wasn't sure if I wanted to do a postdoc, but I ultimately decided I wanted to test myself. I wasn't sure if this was the career for me. I also had a lot of self-doubt whether or not I was actually a good scientist. And I ultimately decided I did want to do a postdoc because I wanted to prove to myself that I could do this, that I was a good scientist, that I could do really, really good research. In the end, I thought to myself, if it doesn't go well and I don't do well in a postdoc and it's not for me, I'm happy that I have a PhD, it was successful, and I was happy with that.
A lot of what you said resonates with me for sure. The imposter syndrome,
Yeah, for sure.
I think probably resonates with most people in science. We're all just constantly unsure if we're good enough, right?
Yeah.
So it's probably really good for people to hear that someone who is clearly successful now has had those thoughts kind of throughout.
And in my experience, the best scientists that I know are the ones that doubt themselves the most. I think it's probably that mental thing that you have, whether you're not sure if you're good enough, actually pushes you to be better.
How do you maintain that edge without the anxiety? Because that's the best place to be, right? Where you're like always trying to be better, but not having the negative emotions tied with the imposter syndrome.
Yeah.
How do you get there?
I think the longer that you're in research, you just see so many different people. There are so many different personalities, people with different abilities, and also different people have different traits that work well. I always think that there's, almost like 20 different personality traits that you need to be in this career. And of course you can't have all of them. But you got to focus on the ones that you do have. Again, if maybe you're incapable of the big picture view or something, but you're really good at generating consistent data, and you have very good ideas, and you've got great critical thinking ability to improve your own data, and maybe you've got a lot of perseverance and persistence. That's another important trait to have, for example, in a PhD. Just you got to keep going and pushing yourself. In academia and labs, the people that I work with are always very supportive. Everybody knows how difficult it is to do this work. And so you got to open up to other people as well. Like if you're not sure of your data, you're not sure you're good enough, talk to people. Everybody's kind of been there, and you'll find support with the people in your lab and colleagues and friends.
Yeah, a good environment is key to success, I think.
Yeah. I feel like the toughest labs, usually the people have a stronger bond. When there's a lot of demands and you're working in a fast-paced, very tough environment, usually I find that everybody's very supportive and work as a team, almost as a family. And that was really important for me in my PhD, was the learning from the postdocs around me. I had really good mentors. For example, Sophie Helene was a postdoc in David Holden's lab. She's a PI now in Harvard. She's doing really, really well. She was fantastic. Teresa Thurston was a postdoc as well in David's lab. She helped me a lot in my PhD. She's also principal investigator as well in Imperial. So these people were really, really good. And without them, I don't think I would have been able to do it. And so I relied a lot on the support of the senior people in the lab. So I think that's an important decision, I guess, if you're choosing a lab to go to, is what are the type of people that are there? Are they going to help you? Are they supportive? It's an important, almost as important as choosing your supervisor is the lab environment, I would say.
Right, especially in a very big lab or fast-paced moving lab where the PI may not even really be around much to
Yeah.
to mentor you. You're
Sure.
really being mentored by those people you see every day.
Yeah.
And so best choose wisely.
I think so. Yeah, I think so.
Fast forward after the PhD, you landed here in the United States
That's right.
in San Diego. How did you end up here?
I ended up here because I was really interested in the research that Richard Gallo was doing. I was working on group A streptococcus, which of course is a very important skin pathogen. So I was very familiar with the literature from Rich Gallo's lab, also Victor Nizet, who had done a lot of really good work on group A strep. And so these were the two big labs that I was familiar with. And I guess kind of in a maybe funny way, working in a very rainy, grey city like London, where my office window looked to another grey wall, I would be on the lab pages of the Victor Nizet lab and Richard Gallo, and you'd see the lab photos of everybody on the beach. I was like, oh, San Diego looks like a very nice place to do research. When I finished my PhD and I thought where I wanted to do my postdoc, I immediately thought of these type of labs. I wanted to work on the skin microbiology. And so I contacted Rich to see if there was an opening for me. And unfortunately at the time, there was no space. Rich was very, very nice. We emailed back and forth a lot. I came to San Diego anyway because my girlfriend at the time also found a position in San Diego. And so I moved to San Diego working in a viral lab for the first 9 months. And so I was happy to kind of change careers, but I kept in touch with Rich Gallo a lot. And an opportunity came up when I arrived in San Diego. My supervisor at the time, who was working on HIV, he accepted a position to go to UC Riverside. He got a position there, and I decided that it's not really going to work for me. I wanted to stay in San Diego.
Good choice.
I think that was a very good choice, yeah. And I got in contact with Rich again and said, hey, I'm here in San Diego, remember me? And we had a good chat in his office. He invited me to attend the journal club seminars and the lab meetings. So I joined for a few months, tried to impress as I could, tried to ask smart questions. And I guess I impressed him because he was happy to take me on and offered me a position. So that's how I ended up here in the Gallo lab.
So what was it about skin that intrigued you?
Not even so much skin. I guess it's strep in general I was really interested in, the host pathogen interactions between group A strep and Staphylococcus aureus. I was more interested in gram-positive organisms. I think probably because I was the only one working on a gram-positive organism in the Holden lab. Everybody was working on Salmonella. So I tried to distance myself probably from gram-negatives and really tried to establish myself as an expert in gram-positives. That's what I wanted to do. That's where I thought I should focus on in my PhD. And so I like the skin microbiome stuff. Actually, this is what really interested me in coming to Rich Gallo's lab. He was pioneering a lot of really cool research into the skin microbiome. And that's kind of what I wanted to work on. I like the idea of working on this new emerging field of microbiome sciences.
What did Rich and you decide that you would work on when you joined the lab?
I would follow a little bit in the footsteps of a grad student, Jim Sanford, when he was here. I would follow up a little bit on the research that he had done for his graduate work, which was working on Cutibacterium acnes, or C. acnes. He was looking a lot at these short-chain fatty acids, these fermentation metabolites that C. acnes produces that cause a lot of pro-inflammatory activity and also have epigenetic modification ability as well. And so he was working on a very limited number of strains, just a couple of reference strains, and Rich was really interested in extending that out and seeing if this is a strain type dependent response. So we wanted to look a lot more into clinical isolates of C. acnes. So my initial aim was really to get swabs of acne patients, also people here in the lab. I swabbed their face and isolated thousands of different C. acnes colonies and essentially made a large library of C. acnes isolates to look at their ability to cause inflammation in keratinocytes. So that was my initial goal really at the start.
And that initial screen was the basis for this publication, correct?
So this was the basis for a publication from last year where we actually used this library of C. acnes strain to see if there's a commensal coagulase negative staphylococci that could inhibit the growth of C. acnes. And so this was a publication in JID last year where we found a Staphylococcus capitis strain that had antimicrobial activity against C. acnes, but its activity was less effective against other commensal species on the skin. So it had somewhat of a selective activity. And so this was a very interesting finding, and we thought this could have good therapeutic activities for acne, which of course is associated with C. acnes.
And so the paper you published just recently in eLife was titled Antimicrobials from a Feline Commensal Bacterium Inhibit Skin Infection by Drug-Resistant Staphylococcus pseudointermedius.
Yeah.
So let's talk about this publication's hot off the press. So what was the rationale behind this investigation?
So this publication, this research was connected a little bit to the Staph capitis project that I mentioned, the one that we published last year. We know a lot in this lab about the antimicrobials and the antimicrobial strains that reside on human skin. So we've done a lot of work on the human skin microbiome. And we've also found a lot of new antimicrobials that are produced on the skin from this work. What was less known was the antimicrobials that are present in the animal microbiome. We had a collaboration with Kate Worthing, who is at the University of Sydney. She's a veterinarian, and she's done a lot of work on the animal microbiome from cats and dogs. And so she was interested to see if whether any of these strains could have antimicrobial activity against human pathogens, but also dog pathogens as well. So she visited the Gallo lab a few years back, and she brought with her her library of animal isolates. And we worked together to test the activity. We did a lot of antimicrobial assays on her animal strains. And we found a couple of very interesting isolates on the skin of cats. These isolates were identified as Staphylococcus felis. And they turned out to be very, very potent at inhibiting the growth of drug-resistant Staph aureus and also drug-resistant Staph pseudointermedius.
What were the major methods you used in this report?
The main method that we used here is HPLC. We were able to generate fractions of the secreted supernatant of the Staphylococcus felis. And from these fractions, we're able to identify single peaks, individual proteins that elute off. And we tested all of these fractions for antimicrobial activity. And so we're lucky that we found a very interesting fraction that had a lot of activity. And we took advantage of a long-term collaboration that we have here in UCSD, the David Gonzalez lab. They are experts in mass spectrometry. And so we sent some of our fractions to the Gonzalez lab for them to analyze it. And they came back with some positive hits. We had a couple of antimicrobial peptides that came out as positive hits. Some of these were phenol-soluble modulin beta class peptides, which we've previously demonstrated to have antimicrobial activity. And we also found a very interesting thiopeptide called microcockin P1 that this Staphylococcus felis produces as well.
Sort of an analogy to explain this technology is that you're kind of starting with a very large haystack with a single needle in it, right?
Yeah.
Too much to look through. It's so big. So you basically separate out that haystack into smaller haystacks, right? And then see which ones are doing the effect that you found with the big one, right? And then once you find that small subset that's able to do the thing, then you kind of can dig through that haystack and find the needle.
Exactly, yeah.
Did you have some experience using those technologies before? Or did you kind of have to learn on the fly?
Yeah, I learned on the fly here. So my initial project here when I started was definitely more research focus, just looking at the basic biology of C. acnes. And then it kind of migrated a little bit more towards, I guess, essentially drug discovery and development. This project kind of reflects that a little bit more. I think my interests kind of diverged a little bit while I was in the lab. So I really like this approach, the needle in the haystack approach. I like that there's a very logical sequence of events, logical sequence of research steps that you can do in order to be able to identify these type of antimicrobials.
So you found the antimicrobial,
Mm-hmm.
and now after this is all published, like I guess the implication is that since this commensal microbe can inhibit the skin infection from the pathogenic strain, that it could be a potential treatment.
Yes, this is what we're exploring now at the moment. And so I guess the issue is obviously drug resistance in these pathogens is a very, very big problem. And it's getting worse because there's not a lot of new antibiotics and antimicrobials that are coming out to market. And that's because I think essentially pharma has ignored the problem. It's very, very costly. It takes a very long time to develop a new antimicrobial, and the problem is resistance develops very, very fast. And so there's a new need to identify novel antimicrobials to fill that gap. The issue is a lot of these antimicrobials, these bacteriocins, these antibiotics, which we find a lot in the skin commensal strains, they don't have a lot of useful therapeutic applications in terms of their poor solubility. They're limited in terms of how you can commercially synthesize them. And so one of the approaches that we thought to make was actually to apply the live bacteria as a therapeutic. The bacteria produce these molecules for us. This is what we did in this paper. We infected the skin of mice with Staph pseudointermedius, and then we applied the live Staph felis organism onto the skin, and we found that the felis was able to outcompete the pathogenic pseudointermedius strain, and we saw less inflammation on the skin. And so what we are trying to do now is formulate the Staph felis strain and apply it to the skin of dogs with atopic dermatitis and pyoderma, these dogs that are colonized with Staph pseudointermedius. And so this is our next immediate aim through collaborators that we have at Texas A&M to do this small trial in dogs.
And the thought is that the Staph pseudointermedius is a driving factor of the atopic dermatitis. So you put the S. felis on there, it gets rid of the driving factor of the atopic dermatitis, and then you hopefully have clear skin.
Yes, this is the hope, and that maybe we can help restore a more stable and healthy microbiome on the skin of dogs by having the felis targeting the pseudointermedius, which is what we know seems to be driving a lot of the inflammation and the disease.
Future directions for probably yourself and for this project are going to be working on this clinical trial, right?
Yeah, I think so.
Are you excited, nervous?
I'm excited about it. I hope it works. We've demonstrated the applicability of using a bacteriotherapy approach, at least in humans. The work of Teru Nakatsuji here, our project scientist, he's really pioneered a lot of this in human AD, where we've taken a Staph hominis human commensal strain and applied it to the skin of human patients with atopic dermatitis, and shown that you can reduce the number of Staph aureus on their skin and improve their disease score. They just published this year the very positive results of a phase 2 clinical trial there. And so I think this is a very powerful approach. It's very new, and I think it could work for a lot of skin diseases that are at least driven in part by microbes such as Staph aureus, pseudointermedius, and also potentially for acne vulgaris as well, which we think is also driven in part by colonization of C. acnes.
So with Teru's work that you mentioned, you have sort of a nice blueprint you can follow.
Mm-hmm.
Do you anticipate there being any challenges with your particular microbe that Teru didn't have with his in the clinical trial you're working on?
I hope not. I guess the main thing is how well this microbe will be able to colonize the skin. But we can probably circumvent that by doing repeated applications. So, for example, the Staphylococcus is found on cat skin, but we don't know how well it colonizes dog skin. So it could be a very transient colonization. Maybe we don't need the bacteria to really kind of stably colonize for too long. So that would be one issue when you use a different organism, is how well it survives on the skin. The main thing that we always have to make sure of by doing a bacteriotherapy approach is to make sure that these organisms that we're putting on the skin could not potentially be pathogenic. We do a lot of tests. We apply the organism on mouse skin alone to make sure that there's no inflammatory effect. We also do experiments on human cells with the secreted supernatant of these cells to make sure that they don't have any significant toxic effect. And we also make sure that these organisms are sensitive to a lot of different antibiotics as well, so that we can always remove that pathogen in case there's some inflammation or there's some toxicity issues.
The benefit of all of this is that you're not creating a drug. You're just using something that's already there and trying to replace the bad with the good. And so theoretically, it should be pretty safe.
Yeah, I mean, of course, there's been a huge amount of work to demonstrate that with Teru's trials in humans. And the hominid strain that they apply is safe and it's effective.
It's very exciting. Definitely, I think some of the future of medicine is microbe-based therapies.
Yeah, I think so too. There's a little bit of reticence for people thinking about applying a live bacteria onto the skin, but these are commensal strains which are already on our skin in the case of hominis. It's just a more rare isolate that has this really unique antimicrobial activity. They should be able to tolerate colonization on the skin. And like I said before, a lot of the problems with coming up with new antimicrobials and antibiotics is a lot of these are very complicated to make. Some of these antibiotics, they're ribosomally synthesized, they have post-translational modifications. So there's a complicated genetic system in the bacteria to produce these antimicrobials. That makes them a lot more difficult to have them commercially, commercial purified extracts of these. So applying the bacteria which naturally produce them is will obviously circumvent that big problem.
What's next for you career-wise? Do you see yourself using this experience with clinical trials to kind of jumpstart your future as a drug developer?
I've been thinking a lot about that lately since I've recently finished my postdoc and I'm now a project scientist. I have been thinking a little bit more about what's next in my career. As I mentioned, I really like these projects that I did here, identifying the Staph capitis, which targets C. acnes, and we're using that as a bacteriotherapy for acne, and now the Felis for the pseudointermedius. So I really like this kind of drug discovery approach, and I think I will probably transition to industry sometime in the future. That's probably where my career lies. Hopefully being able to lead a group in maybe, early drug discovery mechanisms. I think I'll stick with looking at inflammatory and autoimmune diseases. I think these are very important. I'd like to stay involved in skin research as well. And so, who knows? I think a lot of what I've learned here in the Gala Lab, I'll be able to translate that into industry and hopefully learn a lot of new cool techniques as well. And I always like just being open to new ideas and learning new things. So that's the main thing for me. As long as I keep challenging myself, learning new things, I'll be happy.
Do you think it's the fast-paced nature, large amount of funding that kind of intrigues you, or is it maybe the lifestyle that you would have as a manager of a team that doesn't involve so much grant writing but more project development that intrigues you?
In academia, you're a little bit more limited. If you want to keep going in academia, you'll have to write your own grants and become a principal investigator. I'm just not sure if that's for me. It's so competitive. Very few people make it, and it's so tough to run a lab and keep your lab funded in academia. Just not sure if that's the route I want to go down. I would like to be focused, more of my time focused on research, on discovery, And yeah, as you mentioned, maybe not be too focused about where the money comes from, or not being able to utilize this particular technique or this technology because there's no funding. And so I think probably based on a lot of conversations that I've had with a lot of friends of mine who are in industry, I think I'm probably more suited to industry, but I don't know until I go. So we'll see if I like it. And
Do you think you're more of a startup kind of guy or larger established company?
maybe larger established company. I feel, I guess, with the conversations that I've had, I think the startup environment is almost like a postdoc in a way, I think. And also maybe it's a little bit more strict in terms of what the goals are, because as a startup, you have very, very specific goals. You've got to prove that this particular technology works, whatever it is that the company is supposed to do. You've got maybe limited amount of funds and limited amount of time to really show that your idea, your drug, your technology, whatever it is, has to work. And so maybe it's more strict. So I guess I would probably think to target some of the larger companies. But who knows? It depends on the position. It depends on the project.
Yeah, you kind of just got to try it out, and if
Yeah.
it doesn't work,
Exactly.
try something else. That's what we do, right, in science?
Yeah.
If you could go back in time and maybe tell yourself something back when you were maybe a PhD student, what would be maybe the one or two things that you wish you knew that you know now?
I think I probably would have liked to have more belief in myself. I think maybe that's the major thing, and we kind of touched on that a little bit. The imposter syndrome. Yeah, and just have belief in my data, trust in my data. I think maybe that's what slowed me down a lot in my PhD. Just wasn't sure if I really believed the data, and maybe I need to, repeat experiments that really didn't need to be repeated, and just being very unsure about kind of taking the project to the next level. So I think just a little bit more self-belief, because, the best thing to do is just do the experiment. Sometimes I overthink a little bit as well. I'm planning the experiment in my head way too long, where in the same time I could have just done it and found out what the answer is. And so I think that probably would be the two things that I would maybe like to have changed. Just a little bit more belief in myself, and then just maybe the courage just to just do the experiment and see what happens. If it doesn't work, no big deal, because you just try something else.
So with that, I just want to ask you if there's any final things you want to say before we wrap up. I want to be respectful of your time. It's Friday afternoon.
I'm not sure if I have anything else.
If anyone wants to reach out to you, maybe we could provide them your email or something like that.
Yeah, absolutely.
If they have any questions about your science or your path, we could drop your email in the show notes if that's okay with you or something.
Yeah, absolutely. Yeah, every lab is different, every person is different, and whether you're doing a master's or a PhD or a postdoc, they're all quite different experiences. I always offer my support. I'll always try and give helpful advice to people who need it and try to be a mentor. And so I was probably a little bit shy back in the day as well, and kind of maybe I could have asked for help a little bit more, and ask people to look at my data, ask for advice. So I think it's very tough to be in this career, and so it's important to get as much help and support and advice as possible. So of course, yeah, happy to provide my email if anyone wants to contact me, and yeah.
Awesome. So ask for advice and pay it forward. Well, thank you for taking the time to chat, man. This has been fun. I'm looking forward
this was
to
very
it.
good. I really appreciate it.
Yeah, hopefully it recorded. All right, we're done.